Good evening, everyone. Good evening. Welcome to Science on Tap. Thanks for coming. My name is Susan Knight. I work at the University of Wisconsin Trout Lake Station. And Science on Tap is an example of the Wisconsin idea, an idea conceived in 1905 that the boundaries of the university are the boundaries of the state. Our partners in Science on Tap include the Monaco Public Library, Badger chapter of the Wisconsin Alumni Association, and of course our host here, the Monaco Brewing Company. So thanks to everyone. Science on Tap is largely funded by a grant from the Brittingham Funds from the University of Wisconsin-Madison. And remember, there are 4 ways to watch. You guys are all right here now, so that's one way to watch. You can also, it is live streamed at our Monaco Public Library, so you can always go over there if you don't want to be here or if it's too crowded. We also have live streaming for anybody who has a fast enough internet connection at their own home. So if you're off in some Netherlands, you can still watch. And you can also watch any event later because we archive everything and you can watch the entire event later. And then about 2 months after we have the event, our 7th anniversary and Founders Day, we have a lovely cake that we are going to share with everybody. Happy birthday, Science on Tap, and happy birthday, University of Wisconsin-Madison. So I hope everybody will stick around for that at the end. We already cut the cake, so you can't quite see all the letters, but you get the idea. Another thing I want to say, since it is our 7th anniversary, I thought you might like to know where are they now, some of our originators. So if you were at the very first Science on Tap, it was presented by the head of Kemp Natural Resources Station, Tom Steele, and the head of UW Trout Lake Station, that was Tim Kratz. And they have both retired and have gone off to find fun and adventure elsewhere. Tim Kratz is in Duluth, and Tom Steele is in Nova Scotia, which is where he is from. And Hank Rossell, who led us with the UW alumni, the Lakeland Badger chapter of the Wisconsin Alumni Association, he was the backbone of our committee and did so much work for us that we are really discovering how much he did. And he is now living in Virginia. So we are carrying on and very happy to have everybody here and delighted that everybody could make it. So, all right. So remember that our next event will be March 4th, and we will have Patrick Goggin back. You might remember he was here about a year ago, and he's going to talk about gardening in the North Woods with native plants. He is not going to tell you how to grow crocus and daffodils. He is going to tell you how to grow shooting stars and things like that. So it should be a great deal of fun. He is a highly successful gardener over in Phelps. So it really is Northwoods. But tonight we have Dr. Dan Preston. Dr. Preston grew up in Portland, Oregon, and he has an undergraduate degree from Oregon State University. He earned his PhD from the University of Colorado, and then he did a postdoc at Oregon State University.
This is a demonstration.
He is currently
on
on the faculty in the Forest and Wildlife Ecology at the University of Wisconsin-Madison campus. His interest in parasites originated with his interest in amphibians, which have been declining due to disease for some time. He's also been fascinated by the unique life histories of parasites and their often bizarre effects on hosts. Maybe you'll talk about zombie ants or something. Okay, here's your trivia question for Dan. Actually, there are 2 questions here, so bear with me.
This is a demonstration
in graduate school,
of
he spent some
Wirecast.
time teaching science to 4th graders. There's a challenge. He wanted to do an activity involving cockroaches, so he purchased some cockroaches and started breeding them at his house in an enclosure. He had read online that Vaseline supposedly prevents cockroaches from clinging to a surface, so he used Vaseline on the inside of the enclosure to create a barrier for them so they wouldn't escape. He wasn't 100% sure of his lid on his enclosure. Well, the cockroaches reproduced, and they were a big hit with the 4th graders.
Of Wirecast.
But some of the babies started escaping into his house. So much for the Vaseline. His roommate at the time started finding some of the juvenile cockroaches in his house. Dan lied, told her they were native insects coming in from outdoors. Eventually, she found out they were from his cockroach colony, and that was the end of that. She made him get rid of them. Okay, that was story number 1. Number 2, here's another story. He also kept reptiles at the time of the cockroach escape. He had a large carpet python that escaped. It escaped her enclosure one night. The python crawled along a shelf in the living room in the night, knocked over several bottles of wine onto the floor. One of the bottles broke on top of their internet router, knocked out the internet at the house. So here's the trivia question. Did Dan confess that the python had escaped and caused the internet to go out? A, yes, of course he told the truth.
Of Wirecast.
B, no, of course he didn't tell her. No, he did not tell her. He lied. Dan Preston.
Thanks for that introduction, Susan. I maybe shouldn't have told you those stories. I think the message is that I have a problem with lying from the stories. I promise I will try not to lie tonight. And I am still good friends with that roommate, so despite the various organisms escaping from their enclosures, we're still friends, we're in touch, so that's probably a good thing. So yeah, as Susan mentioned, the topic that I want to tackle tonight are parasites, and particularly parasites that inhabit freshwater ecosystems. That's kind of one of my major research interests. And I think when we think about parasites, the first thing that generally comes to mind are that they cause disease, they cause lots of problems. We maybe think about ticks and Lyme disease. We maybe think about swimmer's itch. But I want us to maybe think a little bit more broadly about parasites and perhaps even acknowledge sometimes, in some cases, in some parasites, they might play roles in ecosystems that are perhaps desirable for various reasons. And so I'll leave it up to you whether you think that's nonsense or whether that's perhaps a viable perspective. And so we'll see what your opinions are at the end, perhaps.
This is a demonstration of Wirecast.
So before I dive in, I think I'm going to try to tackle sort of 4-ish themes, and I've got a few notes here to kind of keep me on track. And before I dive in too far, maybe I'll just define sort of what I'm talking about when I say the word parasite, because it's kind of one of those things where maybe you know it when you see it, but defining exactly what it is kind of more challenging. And I've posed that question to a lot of undergrads, and it leads to good discussion because we don't always agree. And at least for the context of this talk, I'm narrowing that definition a little bit. But generally, when I say parasites, I'm thinking about organisms that have a symbiotic relationship, so they're living in very close association, and one organism is negatively affecting the other. And that's sort of a broad definition that can include sometimes things like predators. And so to kind of narrow that, we say that the relationship is intimate and it's also durable, meaning it lasts a long time. So that's one of the distinctions between a parasite and a predator. A parasite has a relationship that lasts a very long time. And there's also different types of parasites, and we also use the word pathogens a lot. And for the sake of this talk, I'm thinking about parasites in terms of things that are bigger than viruses and bacteria, which we often refer to as pathogens. So for parasites, I'm thinking about things like worms and ectoparasites, ticks, Things along those lines are kind of what I'm focusing on. And so
This is a
for sort of this
demonstration
first
of
theme, I
Wirecast.
want to touch on the fact that there is a huge amount of diversity of parasites that we usually don't see, we don't think about, and we often ignore unless they're causing problems. And that diversity of parasites has some really fascinating life histories that are worthy of study and are very intriguing, at least to me. And so, in terms of parasite diversity, there are lots and lots of undescribed parasites. So
This
it's
is a
pretty hard to know
demonstration
exactly
of
how many parasites
Wirecast.
there are out in the world. But one way we can try to tackle this is that we do have data on the number of parasites that infect particular hosts. And when we extrapolate that data, so by that I mean we might have a deer, and we know that on average the deer can support maybe 8 different parasites or something like that. When we sort of extrapolate those numbers, and we also incorporate the fact that some parasites can only infect specific hosts, it turns out that there's something like,
This is a
we guess, in
demonstration
the
of
order of 4 to 5
Wirecast.
times more parasites than free-living animals in terms of number of species. So when we go outside, we generally see the things that are obvious, the birds and the deer and the large organisms that we can visibly see very well. But there's a whole hidden community of parasites inside all these organisms that we generally don't see. And the diversity of those parasites is actually much, much higher than the free-living organisms that we do see. So that's kind of one
This is a
unique thing
demonstration
that
of
we often
Wirecast.
don't realize. And in terms of the thing, and Susan kind of alluded to this, that intrigues me a lot about parasites is that they often are very, very, very well adapted to do really weird and unique things. And in particular, they tend to have, some of them have life cycles where they infect multiple different hosts. And so they need to evolve mechanisms to move between these hosts. And they also need to evolve mechanisms
This
that
is a
allow them
demonstration
to
of
do completely
Wirecast.
different things in these different hosts. And so a lot of parasites are almost like taking 3 or sometimes 5 different organisms and kind of conglomerating them all into one organism. And the parasite can do all these different things. And so to kind of illustrate that, I want to focus some aspects of the talk on what are called trematode worms. And that's a particular type of parasite that I find interesting. They're the type of parasites that cause swimmer's itch. And so I want to focus in a little bit on trematodes and tell a couple stories about trematodes. So first of all, trematodes are worms, and they have life cycles where they often infect somewhere between 2 and 6 totally different host species. And what trematodes do is they start their life almost exclusively in a freshwater mollusk, and that's typically a snail. So most trematodes start out in snails, and this can be an aquatic freshwater snail, a marine snail, occasionally a land snail, but usually an aquatic snail. And inside of the snail, they do a number of different things that are interesting. So one of the things they do is they actually castrate the snail, so the snail can no longer reproduce, and they typically displace all of the sexual organs of the snail. So in other words, the little worm gets into the snail, it begins consuming resources from the host, and the reproductive tissues of the snail essentially go away completely. And when we crack open these snails, we find that sometimes as much as 50% of what would have been snail is actually parasite tissue. So that's kind of like thinking about, say you get infected by a parasite, and this is maybe not bolstering my argument that parasites aren't so bad, but say you get infected by a parasite, and it takes up your right leg, your left arm, and like half of your torso, and all that turns into parasite tissue, but you still are able to survive and kind of keep going on doing your thing. And that's what happens to snails.
This is a demonstration of Wirecast.
And so when that happens, essentially the snail's no longer reproducing, but it is producing parasite offspring. So the parasites are reproducing in the snail. So this whole unit, visually it looks like a snail, but sort of from a genetics or an evolutionary perspective, it's functionally a parasite. It's no longer reproducing. It's essentially just a factory of parasites. And there's a term that we use called the extended phenotype, and that's just basically that this thing looks like a snail, but effectively it's just a parasite at that point. So that's kind of one interesting and cool thing about trematodes and snails. Cool according to my perhaps skewed view. The other few things that are interesting about trematodes and snails, they sometimes cause what's called gigantism. So the snails that get infected actually grow larger and live longer. So in other words, they stay alive to produce parasites and send them out into the environment, and they get even bigger. So not only does the parasite take over sort of the machinery of the snail, it essentially turns it into a bigger and more efficient and sometimes even longer-lived parasite factory. So that's one thing that can happen. And while the trematode is in the snail, it is reproducing asexually. So it's not like a male and a female are coming together. It's essentially producing clonal offspring. So it's just reproducing like crazy asexually. So one worm can infect the snail, and then it reproduces like crazy, producing hundreds or thousands or even millions of offspring inside of the snail. So that's going on. So that's interesting. And what's even more fascinating about that is that these worms inside of snails a lot of them have what's called a caste system. So we think of things like honeybees and ants, and they sometimes differentiate different tasks within a colony. So for instance, you have a queen bee that's producing offspring, then you might have bees whose task is specifically to go off and get pollen, or ants whose task is specifically to get food, and others who protect the colony. Well, trematodes do the same thing. So there are trematodes within snails whose specific job is to attack other potential trematodes that infect the snail. So once an infection is there, another worm might try to infect the same snail, and they're actually soldier worms who essentially attack and kill those. And then there are other worms whose whole sort of role is to just reproduce and create more worms. So there's this colony of worms inside the snail. They're very specialized to do different tasks.
This is a demonstration of Wirecast.
So that's the first sort of life stage of the trematode, inside of a snail. While it's in that snail, it's reproducing like crazy within the snail, and then it's also producing parasites that leave the snail, and they look completely different from the parasites in the snail. So if you look at the ones in the snail, they look kind of like a worm for the most part. If you look at the ones that leave the snail, frankly, I think the best comparison is they look kind of like a sperm. They have a body and a tail, and they swim through the water, and they can move around. So they're producing this life stage that's completely different. That exits the snail. And then what they do is they need to find their next host. And that can be a whole range of different organisms. It's often something like an invertebrate. It could be a mammal or a vertebrate host. It could be something like a frog. It could be a whole range of different hosts. So they go from the snail into this next host. And when they're in that host, they often cause a whole range of different effects relative to what happens in the snail. And the reason they cause a number of different effects at that point is that the final host, for the most part, and there's a lot of variation here, the final host is often something that's a predatory animal. So a common life cycle is they'll go from a snail to something like maybe a bug or a fish or a frog, and then into a predator. And the way they get from that middle host to that last host is that the predator actually eats the middle host. So for instance, a heron might eat a frog, and that frog is infected with a trematode, and then the heron gets infected. Does that kind of make sense? And so that process is called trophic transmission, and that's a really common way that trematodes get between hosts in that life stage. And then the trematode ends up in the bird, or it could be a fox, or a dog, or a human. That animal then essentially, what happens is the trematodes reproduce. That time they do it sexually, so two worms come together, produce offspring, and the eggs go into the feces of that host and then pass back into a water body, and that completes the life cycle. So it can go from snail to some aquatic organism to some terrestrial predator back to snail. And that's a kind of common trematode life cycle. And there's lots and lots of variations on that theme. But what's really fascinating is when they're in that host that is then eaten by another host, they often do a range of things which we refer to as parasite manipulation. And what that term means is that the worms are actually modifying something about that host to make it more likely to get eaten by the final predator. And there's a whole range of different ways that this has evolved. And the thought is that this is actually adaptive for the parasite in the sense that if this parasite is in a frog and the frog is more likely to get eaten, then the parasite has a better chance to complete its life cycle. So a few of the ways they do this, just a couple of examples. One particular system I've worked on involves frogs that are eaten by predatory birds and mammals. And the trematodes cause the frogs to actually grow extra legs and to have deformed legs. And this is something that was actually first observed in Minnesota, and it's since been found in a lot of other places. And when we go to certain wetlands, we might see frogs that have literally 10 legs, and they might be all deformed. And initially our thought was this must be pollutants or something along those lines, but it turns out it's trematode parasites that are disrupting the limb development of the frog. And the thought is that that's actually adaptive for the parasite in the sense that a 10-legged frog is more likely to be eaten by a predator than a normal frog. So that's one sort of example of this parasite manipulation idea. Another example is a trematode in marine environments that affects fish, and it actually insists in the brain of the fish, and it produces chemicals that affect the behavior of the fish, such that the fish actually swim to the surface of the water and do this bizarre sort of flashing behavior that is not seen in uninfected fish. And it turns out that these fish that are swimming to the surface and doing this weird behavior are way more likely to be eaten by a predatory bird, like an egret or a heron or something in the intertidal that's eating these fish. And there's been studies showing that when they're infected, they're much more likely to be eaten just because they do this weird behavior. And that's caused, again, by the trematode. One last example of that is trematodes that infect land snails. And these particular trematodes move into the eye stalk of the snail, and they produce this crazy sort of pulsating behavior of the eye stalk. And it's thought that that catches the attention of birds, and then the birds are more likely to find the snail, eat the snail, and then the parasite will infect the bird. So that's just another example of that. So that's called parasite manipulation.
This is a demonstration of Wirecast.
Those are some of the reasons I'm intrigued by parasites. I probably haven't begun to convince you there's anything necessarily useful or good about parasites. So that's kind of what I want to talk about next. And as I mentioned, we assume parasites cause disease, they cause mortality, they cause problems. And in a lot of cases, that is true. But what we often overlook is that they're doing more than just that. And so a couple of examples of that. One is that because parasites infect multiple different hosts, so I mentioned these trematodes that move between all these different species, we actually can go out and measure the number of parasites in an ecosystem, the diversity of parasites, and it turns out that that is very often correlated really closely with the number of different hosts in the environment. And so environments that have a really species-rich, intact community of hosts tend to have a high diversity of parasites. And often the hosts are hard to measure, but we can go out, for instance, and measure parasites in snails. And if we find that one environment has 40 different parasite species, we know that the host community is really rich. And so there's actually been ideas and sort of this concept that an ecosystem that's actually healthy, one that's in good shape with a really rich community, tends to have lots and lots of parasites. And so we can actually think of parasites in some cases as indicators of the overall health or structure of that community in terms of the number of free-living species. So a degraded system doesn't necessarily have more parasites. So that's kind of one example of a parasite indicating that maybe an ecosystem is actually in pretty good shape. And there are also specific ecological roles of parasites that we might think of as beneficial. And one example of that is there is a parasite that lives all over the world, but there's one in Japan, and it's called a nematomorph worm. So this is not a trematode, but it's another type of worm, a parasite. It's something that's called a horsehair worm, and they can actually get very large, like on the order of that long. And what these worms do is
this is a demonstration of Wirecast.
They infect terrestrial insects for the most part, things like crickets. And they actually have a life cycle that requires them to enter a water body to reproduce. And so what they do is when they end up in a cricket, and the way they end up in a cricket is actually pretty interesting too. They actually infect an aquatic insect, something like a mayfly, that starts in the aquatic environment and then ends up in the terrestrial environment. And then a cricket will eat the mayfly at some point. It could be dead at that point.
This
Ingest the larval stages of the parasite. Parasite then matures in the cricket. And then the crazy part is that it affects the behavior of the cricket so that the cricket effectively commits suicide by jumping into a water body. And then the worm escapes the cricket into the aquatic environment, reproduces sexually, and completes its life cycle. And there's been studies on this where one scientist had a swimming pool in his backyard and these crickets would jump in and then the worms would come out, and he actually measured this. And that's one of the best sort of studies we have on that topic. But what's really crazy about this is that in certain streams in Japan, these crickets that are jumping into the water make up about 60% of the energetic intake on a yearly basis of a particular fish called a Japanese char that's essentially a trout. And this particular fish is critically endangered in Japan. And 60% of its energetic intake is these crickets that jump into the water because they're infected by the worm. And the implication there is that if that worm did not exist in that ecosystem, the number of these crickets that are essentially jumping into the stream and committing suicide would be much, much lower, and the food resources for the endangered fish would be much, much lower. So there's potential that the actual persistence of this endangered Japanese char is dependent on the ecological role of the parasite. And that's something that we probably wouldn't know, or we wouldn't even think about or realize if folks hadn't gone out and done that kind of study. And it turns out that when we cut off that subsidy of crickets, the trout actually switch their feeding behavior to eating benthic insects, so things like fly larvae and other stuff on the stream bottom. And when they do that, that changes the community structure of the invertebrates. It even changes the primary production in the stream. And so the fact that this worm is causing these crickets to jump in the water is changing this whole food web in the stream and potentially helping this endangered fish. So that's an example of a parasite potentially doing something that we would actually value from the perspective of its ecological role. And it turns out those sort of examples are not super uncommon. So I've also worked in streams in Oregon where trematode parasites infect snails, and then a whole bunch of predators actually feed on the free-living stages of the parasite, but very few predators feed on the snails. So in these streams I've worked in, the snails are about this long, fish don't really eat them, not much predators are eating them, but because the parasites are in the snail, the parasites come out of the snail, and then things are actually feeding on the parasites themselves, like caddisflies and other benthic drift-feeding invertebrates. All this energy from the snails is moving into invertebrates and other parts of the food web, whereas otherwise it would be locked in the snail. And so it's changing how energy moves through that food web, and essentially allowing organisms to access resources that otherwise they wouldn't be allowed to access. So that's kind of another example. And the times where we've quantified the roles of parasites in food webs, we found that these sorts of effects are pretty consistent across wetlands. We see them in lakes, streams, especially marine intertidal environments. So parasites are actually an important food web player, essentially, in how that community operates. Two more examples about parasites doing things that are not necessarily bad, depending on your values. One is that parasites in some cases can actually help maintain biodiversity of free-living hosts. And there's a couple of really good studies showing that essentially when we have species and environment competing, there are scenarios where without parasites, the two organisms cannot coexist because competition is really strong and one of them tends to dominate the other. But in some cases, the parasites affect that species that is dominant. They reduce its population, and that allows other organisms to coexist in the same environment. And so that's a term we use called apparent competition. And essentially, it's the idea that parasites, by infecting species that can be competitively dominant, they allow other species to coexist. And so that particular mechanism can lead to scenarios where parasites are actually increasing diversity in the community rather than causing mortality or causing extinction. Extinction is generally pretty rarely caused by parasites. And then one last example that is pretty interesting that's more on a level of individuals. There's an idea that some of you may have heard of called the hygiene hypothesis. And this is an idea that In our modern society, we are very hygienic and somewhat sterile, and we've sort of removed a lot of the microbes and parasites from our environment. And because of that, we're actually more susceptible to being allergic to a lot of things. And there's really kind of fascinating biological and physiological reasons for that. But there is some evidence that certain parasites can actually upregulate aspects of our immune system and make us less likely to have allergies and certain autoimmune disorders. So there's actually a line of research in medicine that is testing the capacity of using helminth or basically worm parasites to treat certain autoimmune disorders, things like Crohn's disease, and a few other things. And the reason for that is because parasites can upregulate certain aspects of our immune system and help prevent things like certain allergies and autoimmune disorders. So that's another potentially a beneficial role of certain parasites. So hopefully you're maybe starting to reevaluate. Parasites are 100% of the time bad, which I think is often the perspective. So now I want to shift gears a little bit towards sort of this third theme of the talk, and that is that when we do see large-scale mortality events from parasites, it's very often because of some sort of anthropogenic or human-caused effect. So in other words, sometimes we have modified something that has led to those parasites having massive effects that maybe they wouldn't have otherwise. And two particular examples of that I want to mention, one in Africa and one here in Wisconsin on the Mississippi River. So the first in primarily Africa, but also other tropical countries, there's a trematode worm that causes a disease called schistosomiasis that's one of the leading causes of illness and morbidity in the tropics. It infects something like 250 million people. And this particular parasite starts in snails, and then it moves directly into humans and then back into snails. And the larval stages come out of the snail, they infect a human, and they generally infect the digestive tract and cause a whole bunch of different problems. So this is a huge, huge issue in the tropics. And it turns out that one of the reasons this problem has increased a lot is that we have modified the habitats where the snails live in a whole variety of ways. And one of the biggest ways we modified the habitats is that we have put in dams and lots of rivers to generate hydropower, primarily in these regions, and probably also to irrigate. And when we put in dams, two particular things happen. One is that the habitat where the snails live changes dramatically, and the water gets much slower and generally more productive. And overall, the habitat for snails improves. So when we put in dams, snail habitat improves, and snails move to new areas, and that increases exposure risk of humans to schistosomiasis. So that's one thing we've done that affects that disease. The other thing that we've done is by putting in dams, we've actually eliminated a migratory predator of snails, which are freshwater prawns. So in a lot of these rivers in Africa where we have huge schistosomiasis problems, the natural snail predators have declined or even been totally extirpated because of the dam construction. So now we have this scenario where you have these slow, nutrient-rich waters loaded with snails. The prawns that previously may have kept snail populations in check have been eliminated because they can't coexist with dams. And so now we have these reservoirs that are just sort of hot spots of schistosomiasis. So the point there is that by altering the environment, we've created a scenario where these particular parasites have become more of a problem than they would be initially.
This is a demonstration
So that's
of
one
Wirecast.
example. The second example is that very often when we move parasites around the globe, unintentionally for the most part, those parasites are then interacting with hosts that they have no evolutionary history with. And when that happens, we can sometimes have massive mortality events of hosts. And that's because we've moved a parasite to a new environment. And so that parasite frequently doesn't want to kill the host because that's sometimes bad for the parasite, because if the host dies, it may not be able to complete its life cycle and continue on with its business. But when we move parasites to new environments, they often infect naive or new hosts. And that's the case with things like the white nose syndrome infecting bats, the fungal pathogen that's killing off amphibians. These are generally novel parasites or pathogens that we move to new places. So when we have these huge die-off events, it's often not endemic native parasites to the region. And one example of that is that in the upper Mississippi, there's a particular snail that's native to Europe. It's called a faucet snail. And we've recently moved, well, we moved that snail to the US actually a long time ago, but something around 15 or so years ago, these snails started appearing in the upper Mississippi in big numbers. And it turns out that when these snails came over from Europe, they brought with them at least 3 different trematode species. And these particular trematode species move from snails into birds. And the birds actually eat the snails, they become infected, the trematodes live in the digestive tract of the bird. And in Europe, because these trematodes and snails and native birds have co-evolved for a very long time, they don't particularly cause major problems in the birds. But when these snails and trematodes showed up in the Mississippi River, they started infecting our native migratory birds in the Midwest. And these birds are not adapted for those trematodes at all. And what happens is when they become infected, they actually die from these trematodes very, very fast. And it's something like an estimated 80,000 migratory birds have died from trematode infections in the upper Mississippi River. So in areas kind of around La Crosse and that region. And this includes things like ducks and geese and mallards and a few other taxus. So they eat these non-native snails, they become infected by the non-native trematodes, and then they tend to die in really large numbers. And so, you know, that's clearly a case where a parasite is doing something that's completely undesirable, but it's also a case where we moved that parasite to this region unintentionally some time ago, and it's created this new scenario. So one important distinction is between native endemic parasites, where they are co-evolved with their hosts, and totally novel parasites, which often wreak havoc and do very different things. So that's 2 sort of reasons why sometimes parasites do cause seriously big problems. So the last sort of theme I want to touch on is swimmer's itch here in Wisconsin, because I'm guessing a lot of you are familiar with that. I've had swimmer's itch. It's no fun. It's gross. It's itchy. So I thought I'd maybe just touch on that, and then we can also talk more about it after the talk. Swimmer's itch is caused by a trematode. So these worms I've been talking about a long time, it's caused by trematodes. It starts out in a snail. There's generally thought to be about 3 or so different types of snails that are associated with swimmer's itch here in this sort of region of Wisconsin. We initially had thought there was maybe 1 or 2 or 3 different trematodes that cause swimmer's itch. Now that we have better ability to differentiate species with molecular methods, there may be as many as 20 different trematode parasites that can cause swimmer's itch. And this is a group of trematodes that we refer to as avian schistosomes. And they're called avian schistosomes because they always infect birds. So these are generally parasites that want to, or they're adapted to move from snails into birds. And they typically infect things like water birds, things like mergansers, mallards, geese, ducks, things that show up on our lakes. So those are the kind of birds that typically are infected by
This is a demonstration of Wirecast.
And what happens is when the snails are infected, a single snail can release thousands of larval trematodes on a daily basis. And this happens generally in summer when temperatures are warm. As many of you are probably familiar with, there's sort of a peak of swimmer's itch infections that are often reported around July and August, and that's when the snails are releasing literally thousands of these things into the water body on a daily basis, one snail. So it could be a very low number of snails infected that are causing a swimmer's itch problem at a local lake. And then what tends to happen is we can have wind movement that concentrates the larval trematodes in certain parts of a lake. So when the surface of the water is moved because of wind, you might have all these larval trematodes ending up in one region of the lake. And if that happens to be where people are swimming, that can be when you have a big problem. So where the other side of the lake might not have many of these things, the beach is maybe where things are accumulating, depending kind of on the winds. And what tends to happen is that the cercari, that's what the larval trematodes are called. They're called cercari. They infect the skin of birds. So when a water bird is in the water, it's swimming around, the trematode moves into the foot or the leg of the bird. It's adapted to move through the skin of the bird into the circulatory system of the bird, and it ends up lodging itself in the veins around the intestine, and eventually it reproduces there, produces eggs. Those go back into the water and infect the snail again. What happens is the larval trematodes infect our skin, not because they're adapted to infect humans, but because we have certain cues that when we come in contact with them, the trematode essentially thinks this may be something I can infect, tries to infect it. The larval trematode burrows into the very top layer of our skin, and it can't get further than that, and then it essentially dies, and that's what creates the little localized swelling and the rash, the little bumps and the itchiness. It's the dead larvae of the trematode. So the trematode, yeah, maybe that's not that reassuring. But what is reassuring is that those trematodes are not adapted to infect humans. They've never really caused problems beyond the rash. Occasionally it's associated with fever and things like that. But they're not going to move into your circulatory system. They're not going to cause the same problems as they do in a bird. So in other words, it's sort of an accidental, serendipitous thing. We're not the preferred host of the trematode. They would rather be finding a foot of a merganser. So maybe that's a little bit of consolation about that. And these particular trematodes that cause swimmer's itch are common throughout the U.S. and Canada. They're very common in the upper Midwest, in Michigan and Wisconsin, but they're found all over the U.S. and Canada and other parts of the world. And then lastly, a little bit about maybe how to control it, but I'm not going to give you any super satisfying answers about that, unfortunately. One approach that's been used is treating with copper, which is essentially a poison that kills invertebrates sort of in a wholesale way. And so there has been historically a lot of copper treatments where we dump copper into lakes and water bodies. And I'm not endorsing or recommending that by any means because it kills everything essentially that's an invertebrate.
This is a demonstration
and so
of
in some places
Wirecast.
that's been used historically. I don't think it's as commonly used in Wisconsin. It is used in some other places. One really fascinating thing that's underway in Michigan is that they're actually catching, the DNR in Michigan is catching mergansers that are thought to be spreading these trematodes. They're relocating them from lakes where they have major swimmer's itch problems, and they're generally taking them to the Great Lakes and to other places to release them. And this is costing thousands and thousands, hundreds of thousands of dollars. But in Michigan right now, you can apply for essentially the permit to do this and get some government support to do it and relocate mergansers. And there's some evidence that maybe that's working to some degree, but it's extremely costly. And some of the problems are perhaps that there are multiple species of trematodes and there may be multiple species of birds that are causing the problem. So if we focus entirely on the mergansers and move them, that may cause some alleviation of swimmer's itch problems, but whether it's a long-term solution and whether it works everywhere, I guess I don't know entirely. But that's something that's underway in Michigan, which is pretty fascinating. Two other possible thoughts are that there are predators that eat snails in our region, things like pumpkin seed and some other sunfish that actually specialize on snails. So I think there could be potential to augment a food web of a lake to increase snail predators and perhaps reduce snail abundances such that that might help with swimmer's itch. There haven't been great studies to kind of demonstrate that will really work well, but I think that's perhaps one promising avenue.
This is a demonstration
and then one
of
other
Wirecast.
thing that's been proposed is sort of figuring out ways to create unfavorable conditions for snails, which can mean things like removing invasive plants and other things that maybe we don't want anyways. Those are sometimes good habitat for snails. So there could be some potential there. But I don't have any silver bullet for swimmer's itch, and it is sort of thought to be native snails and native parasites that are causing it. It's sort of part of our ecosystems in the lakes here. It is a huge nuisance, and I know it can be a problem, and it can affect visitation and swimming and all kinds of other things. So I think it's an area where we need to do more research. But at least right now, I don't have a sort of a silver bullet about swimmer's itch. So yeah, with that, I think I've hit my sort of 4 topics. So maybe I'll stop talking.
Would everybody remember to hold the microphone real close to their mouth so that everybody who's listening online can hear well? And Carol will remind you. I don't think I turned that on.
This is a demonstration of Wirecast.
2 questions. How big are the bugs, and are they more on clear lakes or not?
Do you mean the ones that cause swimmer's itch, or?
Yes.
Yeah. So that's how big are they? So they're very, very, very small. So the worms that are infecting snails are visible to the naked eye, and you can see them very well under a very low-powered microscope. The larvae that emerge from the snails, which are what are infect your skin,
This is a demonstration
are much
of
smaller,
Wirecast.
and you can see them with the naked eye when you sort of hold a vial up to the light, but you kind of have to know a little bit what you're looking for. But you can see them. So they're fairly small, but you will have no real indication when you're at the lake that they're in the water. They're too small to kind of notice for the most part. And then, sorry, what was the second part? Oh, the water clarity. Yeah. You know, that's a good question, and I can't say that we know for sure or have really strong trends about water clarity. What I will say is that there is some evidence that when we have nutrient-rich lakes, it causes snail populations to boom. And when the snail populations boom, that can be good for the parasites. So if we have a really nutrient-enriched lake and we have lots of resources for snails, that can sometimes move all the way up to the parasites and cause more parasites. But in terms of just looking at the turbidity of a lake and knowing if that's a good or a bad lake for swimmer's itch, I don't think we really know. Yeah.
This is a demonstration of Wirecast.
So, listening to your stories about the life cycle of these parasites, I read a lot of science fiction, and it just sounded like science fiction.
Yeah.
Oh my gosh, the stories you could write with these insects. My gosh.
And the movie Aliens, you know, when the alien comes out of the person's chest, you're like, yeah, that's not something that's actively happening. But like within the realm of parasites and animal interactions, like, yeah, some of that stuff that's kind of crazy. It's like, yeah, they've actually evolved to do some super crazy stuff. And that's what I find intriguing about them, and also why they're so useful to study from sort of a biology and an evolution perspective. And when we learn about these things, we learn about basically how unique traits and adaptations evolve in the animal world that are really worthwhile to understand. Yeah.
Question way back here.
I'm just curious how much we know about like the mechanism when like a parasite takes over a grasshopper.
This is a demonstration of
and then it
Wirecast.
changes the grasshopper's behavior.
Yeah.
How exactly does that happen? Is it just a nervous system thing or what?
Yeah, it's a great question. And in that particular case, I don't think we've worked out the mechanism. So we know that it happens very clearly, and we have the data to show that, yes, when they're infected, they exhibit this behavior that normal grasshoppers, may occasionally fall into a stream, but they're not actively seeking out a stream and jumping in. So it's a very clear behavioral manipulation.
This is a demonstration
in the case
of
that I
Wirecast.
mentioned before with the trematodes that infect the brains of killifish and cause them to exhibit these strange behaviors, there actually has been some of the very fine-scale physiological and molecular work to show that they actually do modify the brain chemistry of the fish in a way that affects their behavior. So there's other parasites like toxoplasma is one some of us have maybe heard about, and that's sometimes that's associated with risky behaviors. And this is another case where there are mechanisms at play where the parasites are essentially affecting pathways in the brain. And I can't say that we have figured out all the detailed mechanisms, but at least for the trematodes and the killifish story, they are thought to be secreting some sort of chemicals that are leading to this effect. In other cases, it's not necessarily a biochemical effect. Like with the frogs that grow extra legs, that's for the most part, it's just that the trematodes are insisting around the legs of the frog, and as the limbs grow, they are literally hitting the cysts of the worm, and that's causing the cells to differentiate improperly, and it leads to these multiple legged scenarios. So in that case, it was thought maybe that's a growth hormone or something, but it doesn't seem like that's happening. It's just a physical sort of thing. So this has evolved multiple times in this group of worms, and sometimes we kind of know the mechanism, but in other times we're still working on it. It's kind of hard to tease out.
So you were talking about the trematodes sometimes attacking specific hosts or how they find their host.
Yeah.
Along those lines of chemistry, is there a lot of chemistry that goes into how that trematode finds a host, or is that how do they detect them?
Yeah, that's another really, really great question and an area of active research. I think we haven't entirely settled that one in the aquatic environment. There's some evidence that they are able to pick up on chemical cues and potentially move or actively seek out hosts through waterborne cues. In other cases, it seems that it's a semi-random thing where the parasites release thousands of larvae. They kind of are doing this random thing in the water column. They also only live about 24 hours, so their investment is produce as many as we can, and then randomly hope some of them bump into hosts, and then they will be able to kind of complete their life cycles. So I think depending on the specific trematode, there's some evidence where they maybe have some ability to select or detect hosts. But I think more often it seems like they just produce as many as they can, send them out there, hope they bump into the right things. But there's also, that said, there's a lot of variation in this group of parasites. There's a handful of trematodes that actually go from a snail, they come out of the snail, then they're eaten by a fish, and what those trematodes do is they ball up together and they're attracted to light, and they sort of move towards the water column, higher in the water column. They ball up and they do this behavior that makes them collectively look like a larger prey item to a fish, and then a fish eats the whole ball. And so that's like hundreds or thousands of them that come together. And so that's a very adaptive thing to attract a host. And those ones have very long tails, and they're adapted to look like something tasty to a fish. But in other cases, it seems like it's more of a random thing. But in this group of parasites, yeah, they're just doing all kinds of different things. Yeah.
Has researchers or scientists or doctors infected somebody with a parasite and cured their autoimmune disease? Or just
that's a great question. So my understanding about that line of research is that lots of research is ongoing, and it's not entirely my wheelhouse, but I think they're at the point where there's enough evidence that these sort of effects are possibly happening, that we are in the phase where we are doing studies on this, and there are studies where people are infected with worms who have autoimmune diseases, and we're looking at sort of their long-term trajectories. And I'm not sure it's at the stage where that's something that is commonly prescribed in hospitals right now, although it may be in some parts of the world. I'm not actually positive about that, but it's more in the research stage, but it seems promising. There have been studies showing that these worms may offset some of these autoimmune disorders, which people can deal with for a lifetime and cause really major problems. So it's potentially a promising thing, but we're not quite at that stage where it's something where I think doctors in the U.S. are prescribing at the moment.
And one more, and are eagles carriers?
Our eagles, yeah. So eagles are carrying all kinds of different parasites. So things that, in general, predators are loaded with parasites that they're receiving from their prey. So things like predatory birds that are eating fish are very often loaded with all kinds of different parasites in general. I don't know about specifically things like swimmer's itch parasites, but yeah, eagles are, if you were to open an eagle, it would be loaded with parasites. Yeah.
Other than the swimmer's itch, should people be concerned about any other parasites in the water or eating fish? Or
this is
that's a good question. In general, I tend to cook my fish pretty well as a safety precaution. I'm trying to think if there's any particular other parasites around here. There are potentially contaminants in fish that we should be worried about. But if you cook your fish, you're generally not going to have any problems with consuming parasites that can infect you. There are a couple of interesting cases, or unfortunate cases, I guess, where people have eaten undercooked frog legs, and then they have actually acquired some parasites that typically would infect something like a canine or something like that, and they've ended up in a human and become infected, and that can be a problem if you eat really, really undercooked fish. You might or frogs or something like that. You might end up with something that generally doesn't typically infect humans. But if you're cooking your fish and your other meats, then it's not generally a big concern.
This is a
Is chronic wasting disease at all associated with a parasite?
Oh, that's a good question. So chronic wasting disease is caused by something called a prion, which is generally not thought of as a parasite, but it's actually it's a very bizarre thing, and it's a protein that is misfolded, and it essentially causes other proteins to become misfolded. It's a transmissible infectious protein. That's what a prion is, and there's a few prion diseases out there. And so I wouldn't call those parasites necessarily, but they would be called an infectious agent, which is sort of a broader level term. So it's not a parasite in the sense of a worm or something like that that I've been talking about, but it's another type of infectious agent that's essentially just a protein, which is kind of crazy. Yeah.
Earlier, when you were talking about those snails that are invasive on the Mississippi, faucet snails, I believe.
Yeah.
Are those kind of coming in because there's less of the native snail species, or are they just out competing? Could you potentially get rid of that parasite by reintroducing some of these native snail species and pushing out the invasive ones?
Yeah, that's a really good question. And there's some folks at UW-La Crosse and some other folks at USGS working on this issue, and we're just starting a little bit of research on this issue. And I think the state of the knowledge is we're sort of mapping and trying to understand where these invasive snails are and what factors are promoting their abundance. But my sense is the Mississippi is such a huge system that eradicating those snails at this point is probably going to be really, really difficult. It's likely they are competing to some degree with native snails, but that could actually be a scenario where the fact that we have flow regulations through these huge dams on the Mississippi is potentially improving habitat for those snails. I haven't really seen great evidence of that, but the Mississippi is, it's managed for a waterway for commerce and travel, and so it's now a series of very big, sort of slow areas, and some of those areas make pretty good habitat for snails. I think there's some evidence those faucet snails might be limited by temperatures, and that there's some areas where groundwater is coming to the Mississippi that in the very cold winter months, those might be areas that are kind of hot spots for the snails. But yeah, the short answer is I think we're still kind of trying to figure that out, and they're probably here for good, unfortunately. Yeah.
Can you repeat that?
Oh yeah, what do snails eat? So snails, for the most part, most snails are grazing on hard surfaces or plants or other things like that. So they're eating things like algae attached to surfaces. They're eating detritus, which is essentially just organic muck on the bottom. They might be eating organic or decaying matter like wood and stuff like that, but mostly they're eating, things like algae and bacteria and fungi that are attached to surfaces. Yeah.
Viral hemorrhagic septicemia. There was a large die-off of fish on Lake Winnebago a number of years ago. It's supposed to occur every 6 or 7 or 8 years.
Yeah.
Second question is, Shawano Lake has a lot of snails in it.
Yeah.
They had quite a die-off of coots, mud hens, whatever you call them, 7, 8, 9 years ago.
Yeah.
And they literally netted them all up on pontoon boats and had piles of them and buried them.
Yeah.
And Shawano Lake has always been known for waterage. Does this, the first question about viral hemorrhagic septicemia, does that have anything to do with parasite stuff?
So that is caused by a virus, and so, it's, in my thinking of parasites, I often distinguish viruses and bacteria from macroparasites, or these worms and things I'm talking about. So it's caused by a virus, which is essentially could be thought of as a parasite, but it's sort of a bit of semantics whether or not we think of that as a parasite. But it's caused by a virus. So that's a really tiny organism that's kind of doing its whole other weird lifestyle, where it's sort of co-opting the machinery of the cells within the fish and replicating itself, and it's a whole other class of organisms. So it's pretty different from the types of parasites I'm talking about, but it's another infectious agent. So I used that term earlier for prions, viruses, pathogenic bacteria, parasites. All those are infectious agents. So it has some similarities. Regarding the other question about the lake, I'm not specifically familiar with that particular lake, but thinking about waterbird die-offs, there's two things that come to mind. One is something called avian botulism, which has been a problem for waterbird die-offs, and that's another pathogen that causes a lot of problems in certain areas, and it's associated with algae and conditions that are often nutrient-rich. And then the other thing is it's possible that that's a faucet snail trematode related thing. But my understanding is, I don't think we've seen that in a lot of lakes in Wisconsin. It's been more in the Mississippi, and some lakes in Minnesota have shown it. But it's possible that that's this particular parasite. I'm not sure about that lake, but we could probably look into it and find out.
Wait.
Oh, then maybe it is. Then maybe it is this case with faucet snails and these trematodes from Europe. If they found that out, then it sounds like this could be one of these cases where those birds are eating these non-native snails, getting sick with the trematodes and dying. Yeah.
We've got a question online with Susan.
You talked about amphibians being in trouble. Are they in trouble all over the world? Is it worse in temperate regions or is it worse in tropics?
Good question. I would say in general it's worse in the tropics. So the amphibians, I mentioned that we sometimes spread parasites and pathogens around the world. The number one, at least disease-causing problem for amphibians is something called the amphibian chytrid fungus, and that's a fungus that is thought to probably have originated in Africa, and we've spread it all over the world. One of the interesting ways we may have spread it is that back in the 60s or so, we used frogs as a pregnancy test for women. We would actually inject the urine of a woman into a frog, and the hormones in the urine would cause the frog to ovulate. And so because of that, we moved these frogs all over the world, and they were kept in hospitals, because at the time that was like the state of the knowledge for determining if a woman was pregnant. And it's thought that maybe that particular frog had spread this chytrid fungus all over the world. There's some evidence of that. We're not 100% sure of that. But the frogs in the tropics, getting back to the question, are dying of this chytrid fungus at high rates in very pristine environments, in streams, in mountain areas, areas where habitat is great, and it's because of this fungus that's killing the frog, and it's caused dozens of extinctions of frogs in the tropics. So I think that's probably where amphibians are most hard hit. Around here, it seems like a lot of the amphibians are more tolerant of the diseases, but things like habitat loss and invasive species and other factors are playing a role in declines. There are some amphibians in very high elevation mountain lakes that are also susceptible to this chytrid fungus, but it seems like in Wisconsin, it's not quite as much of a problem maybe as it is in some of these other areas like the tropics and other regions where it's actually caused extinctions of amphibians.
Around the Wolf River, they've never heard of anybody getting swimmer's itch on the rivers, but on the lakes right around it they do. Is there a reason?
Yeah, that's a great question. And my hunch is that these particular avian schistosomes, which are the trematodes causing swimmer's itch, are not adapted to living in lake or in river conditions. And so in other words, there are specific trematodes that live in rivers, and they have different mechanisms for finding hosts that allow them to deal with flowing water. So like, for example, there are some of these trematodes we study in Oregon, and they actually don't have tails, and they inchworm around on rocks, and that's an adaptation to living in a high flow environment. So perhaps these particular avian schistosomes are just not found in rivers, which would make sense if we're not getting swimmer's itch in those areas. Maybe in really, really slow flow environments you could see that, but it seems like it's primarily more of a lake and wetland problem because these trematodes tend to live in those particular habitats, is my hunch. But I think you're right. I don't really hear of swimmer's itch as a problem in rivers for the most part. Yeah.
Any other questions, or you all want cake?
Yeah, free cake.
Can you tell me when swimmer's itch was first recorded or founded or people documented it?
Oh, you know what? I don't know the answer to that, but my guess is in Wisconsin it was first recorded or noticed when the first settlers moved to our area and went swimming. That's my guess. I don't know. But we do see an increase in the reporting of swimmer's itch, but it's not entirely clear if that is because we are reporting it more or it's actually increasing. So it's, like I said before, it's thought that it's caused by parasites that have probably lived here. In other words, they're native parasites, and they seem to be native snails. Yeah, but there
This
are
is a
some reporting
demonstration
mechanisms
of
online where we're trying to
Wirecast.
keep track of this, and I would encourage those of you who have swimmer's itch problems to look into those reporting mechanisms. And we have a project that's kind of in the proposal stages where we're hoping to develop more of a reporting framework in Wisconsin to get a better sense for where it is and what kind of habitats are tend to promote it, just to kind of keep better track of it.
Well, thank you very much. Thanks to Dan Preston. Thank you.
This is a demonstration of Wirecast.
Thanks also to the Minocqua Brewing Company, our host, and to our other sponsors, the Lakeland Badger chapter of the Wisconsin Alumni Association, Trout Lake Station, Minocqua Public Library, and Kemp Natural Resources Station. Remember, our next Science on Tap will be March 4th, and we'll have Patrick Goggin talking about native gardening in the North Woods. And we've got cake, so don't go away. And happy birthday to the university, and happy birthday to Science on Tap. Thanks again for coming.
This is a demonstration of Wirecast.